Okay, so ladies and gentlemen, good morning. So today we are going to look at the DNA replication, and don't forget to subscribe and join the YouTube community. So introduction to DNA replication. DNA replication is a fundamental process that occurs in all living organisms. So generally, this DNA replication is taking place in every living organisms because there is no living organism that doesn't have DNA. So usually organisms grow, develop, and survive, and also maintain their life processes through this DNA replication. So it is very fundamental in every living organisms. And DNA replication, it ensures that genetics information is accurately copied and fast during cellular division. So ladies and gentlemen, if you notice, you will realize that the child and the parent look similar. And this is actually because of the DNA replication, because the information are copied from parent to their offspring during the cellular division. And this cellular division is usually taking place, especially meiosis, during fertilization. So that is where we look like our parent, we look like our brothers and sisters. All this is actually due to the DNA replication. And this DNA replication is crucial for growth, development, and maintenance. So we grow and we develop and we maintain our body system all as a result of DNA replication. So that is why whenever there is a DNA, when there is a problem in DNA replication, so there will be a problem in our growth, in our development, and our body maintenance. And that is why sometimes we easily get aged, we die, we come up with a lot of different diseases, especially genetic diseases. And it's actually as a result of problems in DNA replication. So in a DNA replication, ladies and gentlemen, a simple way, it is actually a process where a molecules of DNA replicated itself. When you have one DNA, then it will now replicate itself into two. Like for example, you can see clearly from here that this is actually this is the DNA. So after it actually replicates, it will now produce a two copies. So a DNA replication is a process whereby a one copy of DNA multiply itself. And that is what we call DNA replication. So as we can see from one, it's now turned into two. So this is what we call DNA replication. And this DNA replication, according to Mark Delbrock, he suggested that there could be three possible ways in which DNA could be replicated. The first one is actually semi-conservative. What is semi-conservative? A semi-conservative is actually a DNA replication where two copies of double-stranded DNA, each containing one old strand and one new strand. So look at it from here. You see that this is actually the DNA. So in a semi-conservative, you see we have two copies of the DNA. So if you realize that in the two copies of the DNA, DNA has two strands. So in the semi-conservative, one of the strand is new and the other strand is old for the two copies of the DNA. And that is why it is called semi-conservatives. You see, this is the old DNA. So you see, this is one of the DNA with double strand, and this is also another DNA which is double stranded DNA. So you see, one is old and one is new. Also in this case, one is old and one is new. So that's what you call semi-conservatives. Then in the case of conservative DNA replication, so the DNA replication in conservative produces two daughter double-stranded DNA, one of which consists of two original strands. You see, one of the DNA contains the two original strands, whereas the other daughter DNA consists of two newly synthesized DNA. And that's why here we see that we have two newly synthesized DNA in the daughter cell. So we have two newly synthesized strands in the daughter DNA. So that's what you call conservative. Then the next one is actually the random dispersive. In the case of random dispersive or dispersive, the DNA replication in which the original double-stranded undergoes fragmentation, and then the fragments synthesize complementary structure, both of which assemble to form two replicas. So you see, if you can see clearly here, you can see that in the case of this passive, there is a random combination of newly and old DNA strand. So this is actually basically the three different types of DNA replication that could actually take place in a cell. So then the next thing is actually, what is the importance of DNA replication? So accurate DNA replication is essential for genetic fidelity. You see, some people actually are coming off with genetic diseases like hemophilia, baldness, sickle cell disease due to inaccuracy in DNA replication. But DNA replication is very, very important to make sure that there is a genetic fidelity across generation. It also prevents mutations that can disrupt normal cellular functions. So during DNA replication, it ensures that the mutations are prevented in order to prevent the disruptions of normal cellular functions. Like, for example, in the case of sickle cell anemia, is because of a mutation in one of the nucleotide in the genes that lead to the coding of different amino acids instead of coding glutamic acid, then the value will be coded, which is a non-polar amino acid that lead to the change in the shape, the hemoglobin to sickle cell shaped. And instead of the hemoglobin to be binding with 4 molecules of oxygen, it will be binding with only 1 molecule of oxygen. So DNA replication is very important in ensuring that the mutations are prevented to prevent genetic disorders. So proper replication is vital for the formation of gamete in sexual reproduction. Like, for example, during replication, usually during sexual reproduction, there is fertilization between the egg and the sperm cell. So during that fertilization, meiosis takes place, and the DNA replication also takes place. Genetic recombination also take place. So the most important thing that you need to understand, ladies and gentlemen, in that DNA replication is that the genetics information are copied from parent, recombined during the sexual reproduction. So it is very, very important for the passage of genetics information across generation. So then the structure of the DNA. So the DNA is composed of two strands that form a double helix. So generally DNA is double helix and it is made up of two strands. And each strand is made up of nucleotides, and the nucleotide consists of a sugar, phosphate group, and nitrogenous base. The complementary base pairing is that A pair with thymine and C pair with guanosine, and is critical for accurate replication. Because in a DNA replication, when the DNA are synthesized or is synthesizing, proper nucleotide will be added to complement with the correct nucleotide. So there will be a proper base pairing in DNA replication, and that is what will actually maintain the accuracy in DNA replication. So you can see this is a double helix DNA structure. See, this is one strand here, and this is also another strand here. So we'll realize that from here you have, this is guanine pair with this is cytosine pair with guanine, adenine pair with thymine. So this is the base pairing that lead to actually the formation of two strands. That is the double helix structure of the DNA. So the next thing that we are going to look at is that, ladies and gentlemen, is replication part. This is very, very important on DNA replication. So generally, if you have the DNA for the replication to take place, there must be formation of replication forks, where both the ligand and the leading strand will be exposed. So the replication fork is the area where the DNA double helix is unwound. DNA double helix will unwound itself. So when you have unwound DNA structure, that is what we call replication fork, and it is formed by the action of helicase. So helicase is an enzyme which separates the two strands of the DNA that lead to the formation of replication forks. So you see, this is actually a replication fork, and you can see clearly from here, this is actually the helicase enzyme from here, and it is trying to separate the two stranded of the DNA, and that's how it will lead to the formation of the replication forks. So if one of the most important enzymes in DNA replication, ladies and gentlemen, is helicase, and it's an enzyme that unzip the double-stranded DNA and separate them into two. So this box is served as a site for new DNA synthesis. So it is actually a site for the new DNA synthesis. So therefore, replication forks must be produced, must be generated before DNA replication initiates. So what are the key enzymes in DNA replication? So there are several enzymes that play crucial roles in DNA replication, and this includes the helicase. That is, a helicase is an enzyme that unzipped the double-stranded DNA. The DNA polymerase, which actually aids in adding nucleotides. The DNA ligase, which is actually involved in ligating Okazaki fragments. That is the small fragment of the DNA synthesized. There is also topoisomerase. The topoisomerase is an enzyme that actually remove the DNA supercoiling. Topoisomerase. The topoisomerase is an enzyme that remove DNA supercoiling. Because, ladies and gentlemen, one thing that you need to understand, when the helicase enzymes unwind the double-stranded, as it moves, separating the double-stranded DNA, the DNA will be supercoiled. It will be supercoiling itself. So how do you remove that supercoiling? Is by using the topoisomerase enzyme. So then the DNA polymerase is important in synthesizing new strand of the DNA by adding nucleotides, and that is actually the functions of DNA polymerase. So it synthesizes new strand, while the DNA ligase connects the Okazaki fragment on the ligand strands. So you see, here we have actually talked about the importance of 4 enzymes in DNA replication. Number 1, we talk about helicase. A helicase is an enzyme, look at it, that unzip the DNA, leading to the formation of replication forks. And after that, then the next thing is DNA polymerase. And the DNA polymerase, look at it here, is the enzymes that synthesize the new strand by adding nucleotides. And then, so you see, this is the replication forks after the helicase enzymes unzip the DNA, and then the DNA ligase is an enzyme that connects the Okazaki fragment from the ligand strand. So this is a ligand strand. So generally, the DNA polymerase will not actually add the nucleotides in the ligand strand. Rather, the DNA polymerase will be synthesizing small fragments, and those fragments are called Okazaki fragments. And the DNA ligase will now be connecting those Okazaki fragments, and that will lead to the formation or synthesizing the newly strand, or the newly strand of the DNA in the ligand strand. And of course, as we can see, this is where we have the free nucleotides that are used in the synthesis. And very importantly, ladies and gentlemen, as this helicase enzymes is actually unwinding the DNA, this part of the DNA will be supercoiling. It will be supercoiling because it's like a rock. You are trying to open it up. So as you are trying to open up, or you are trying to separate the rock, other sites, or the two different sites where the replication force is not actually generated, there will be supercoiling. So that supercoiling is resolved by topoisomerase enzyme. So now the leading and lagging strand of the DNA. So generally, actually, as the helicase enzyme separates the DNA into two strands by the helicase enzyme, a leading and the lagging strand are formed. So this leading and lagging strand of the DNA, so the leading strand is synthesized continuously in the direction of the replication force. So it is actually synthesized continuously in the direction of the replication force. So there is no issue. The DNA polymerase will just be continuously adding the nucleotides, and that will actually lead to the synthesis of new DNA strands. So you see, this is where we have, this is the lagging strand, and this is the leading strand. So in the leading strand, there is no issue. Actually, there is no any issue. The nucleotides will just be adding by the DNA polymerase. So you see, first overview, when you have the DNA, the enzymes called helicase enzymes will separate the two double-stranded, and that lead to the formation of replication bubbles. And that is what we call an origin of replication. An origin of replication is a point where the DNA polymerase bind and synthesize and initiate the synthesis of the new strands. So the leading strand is a continuous strand where the DNA polymerase will just be adding the nucleotide continuously without any issue. But in the case of the lagging strand, it's synthesized in short fragment called Okazaki fragment. So it will be synthesized shortly. Small fragment will be synthesized. You see, this is one fragment, this is another fragment, and this is also another fragment. So it will be synthesized in short fragment, and those short fragments are joined together, are connected together using the DNA ligase. And when there is any gap, there is a space where some of the nucleotides are not filled up, then the DNA polymerase 1 will now fill that gap with the nucleotides. So then the initiation of DNA replication, the DNA replication begins at a specific locations called origins of replication. So origins of replication is a site or is an origin where the DNA replication begins. So there is an initiator proteins, and these initiator proteins recognize this region and bind to them, facilitating the unwinding of DNA. So this unwinding creates replication bubbles, and these replication bubbles that expand as a replication proceeds. So as the replication proceeds continuous, then the replication bubbles will also be expanded. You see, this is actually the origin of replication. This is actually the replication bubble. So this is a region, or this is a site where the initiator proteins bind, and the DNA replications begin. And as the DNA replication begins, the replication bubbles will also be continuously expanded. So then primase and RNA primers. So primase is an enzyme that synthesizes short RNA primers that provide a starting point for DNA replication. So generally DNA replication, it doesn't actually occur just like that. The DNA polymerase cannot just start synthesizing new strand of the DNA without an initiator or without some fragment. You see, like, look at this. This is actually a primase. So a primase is an enzyme that make RNA primer. That is a short nucleotide. And these short nucleotides, they are called primers. So these primers are essential because DNA polymerase can only add nucleotides to an existing strand. And if there is no an existing strand, the nucleotide cannot be added. So that is why the RNA primers are synthesized, joined, and then the DNA polymerase will now begin to add new nucleotides. So after replication, the RNA, after the replication is completed, these RNA primers are removed and replaced with DNA. So it's not that actually after the RNA primers are synthesized, joined, and then DNA polymerase initiated, they're not actually going to stay there. They will be removed at the end of the replication. So then what is the DNA polymerase function? So the DNA polymerase is the enzymes responsible for adding nucleotide to the growing DNA strand. It works in a 5' to 3' direction, synthesizing new DNA in correlation with complete DNA. So if there is adenine, then it will add thymine. If there is guanine, it will synthesize it will synthesize or it will add cytosine. So that is what actually happens. That is what the DNA polymerase does. It will just keep continuously adding nucleotide in 5' to 3' direction. So the DNA polymerase also have proofreading abilities to correct errors during synthesis. So sometimes as this DNA polymerase works, it may add some unwanted nucleotide at some point. So after the synthesis of the DNA, at the same time this DNA polymerase has a proofreading activity, it will go back, check where there is a mistakes and correct the mistakes. Then the next thing now that we are going to look at is actually the role of sliding clamp. The sliding clamp is a protein complex that holds DNA polymerase in place during replication. It enhances the enzyme's processability, allowing it to synthesize long stretches of DNA. So you see, this is the sliding clamp. So the major work of this sliding clamp, and this is the clamp loader. The clamp loader, and this is actually the sliding clamp. So the sliding clamp, and this is actually the DNA polymerase enzyme. The sliding clamp is actually a complex proteins that holds the DNA polymerase in place, which actually allow the DNA polymerase to synthesize a long stretches of the DNA. So this mechanism is crucial for maintaining the speed and efficiency of DNA replication. So, ladies and gentlemen, this is actually a brief introduction of the DNA polymerase. So this is actually a brief introduction of DNA replication. And I hope you will find this video very interesting. And please don't forget to subscribe and join the YouTube community. So this video is made easy for you to understand the role of each and every enzymes, the ligand strand, the leading strand, how you generate Okazaki fragments. So I hope you'll find this video very interesting and see you in my next video. Bye bye.